Hello, everybody. Bacterial ribonucleoprotein bodies, also called as BR bodies, are liquid-like condensates that organize mRNA decay in bacteria. They are induced under stress conditions similar to P bodies and stress granules in eukaryotes, and they confer stress tolerance to bacterial cells. The main scaffolding protein of BR bodies is also conserved in pathogens, and they form BR body-like structures. So it is important to study the proteome of these BR bodies, which will provide insights into their assembly and regulation, and eventually will help to generate next generation antibiotics against these pathogenic bacteria. RNA, the major endonuclease in bacteria, is the central scaffolding protein of these BR bodies. It has this structured catalytic N-terminus through which it forms a tetramer, and a highly disordered CTD with a continuous stretch of positive or negative amino acids through which it self-assembles. And at physiological concentration, it requires multivalent RNA molecules, which enhances the self-assembly process, leading to the formation of condensates as shown here. RNase undergoes condensation only in the presence of RNA. The only disordered C-terminus domain undergoes condensation, but not the only catalytic N-terminus domain. In the cells, when this disordered CTD is deleted, BR bodies are completely dissolved. It's well known that RNase forms a multi-protein complex called RNA degradosome, through which it degrades majority of mRNA molecules in the cells. And it also interacts with several other proteins. All these interactions were reported not in the context of liquid-liquid phase separation. So it would be quite interesting to see if RNase interacts with these proteins in BR bodies, or it has completely different set of proteins. So to find that out, we isolated proteins present in these BR bodies by means of enrichment through differential centrifugation. And the protein isolated from these enriched BR bodies was subjected to mass spectrometric analysis. And it was then compared to the proteins isolated from cells which doesn't form any BR bodies. Here is the mass spec data. There are about 148 proteins that are enriched twice above the cutoff value. Importantly, most of the known interacting proteins are enriched in this data set. Cell wall proteins and the membrane proteins, which could be the common contaminants during the protein isolation, are highly depleted. The proteins associated with other biomolecular condensates are also depleted, suggesting that the samples were not contaminated with other condensates present in bacteria. Interestingly, these enriched proteins are quite diverse. There are not only mRNA decay factors, there are also transcription terminators, ribosomal proteins, protein chaperones, and even carbohydrate metabolic enzymes, suggesting that BR bodies could play a role beyond mRNA degradation. We purified a set of proteins from these enriched proteins, including some lowly enriched proteins and some negative factors, which are not supposed to present in BR bodies based on in vivo analysis. And then we tested if these proteins can directly be recruited into BR bodies in vitro. Briefly here, RNase CTD plus RNA condensers are preformed, and then the client protein labeled with sulfide was added. The level of recruitment of the client proteins was quantified based on the partition coefficient. That means the partition coefficient is directly proportional to the level of recruitment of client protein. Most of the proteins enriched in mass spec data are also enriched in the in vitro recruitment assay. This includes the core degradosome proteins and a protein chaperone, a fatty acid metabolic enzyme, and HFQ and RNA chaperone. There are these three proteins. Even though they are enriched in mass spec data, we did not see their enrichment in in vitro recruitment assay. They are on par with negative control, suggesting that these proteins may require other protein factors for their recruitment into BR bodies. Interestingly, these two proteins here, ribosomal protein S1 and S-adenosylmethionine synthetase, dissolved BR bodies in vitro. We concentrated on ribosomal protein S1 because it has been shown to regulate translation and mRNA decay in bacterial cells. To see if this dissolution is physiologically relevant, we overexpressed S1 protein and analyzed the number of BR bodies in comparison to a strain that expresses only empty plasmid. There is drastic decrease in the number of BR bodies when S1 protein was overexpressed. Actually, S1 protein dissolves RNase condensates when they are induced with RNA, but not when RNase was induced with another protein called PNPase. And it's well known that S1 protein strongly binds to mRNA molecules, and we did not see any direct interaction between RNA-Z and S1 in the pull-down assays. The first line here is the positive control, and these are the negative controls. These results suggest that S1 protein dissolves BR bodies by sequestering RNA from RNA-Z. And then we tested if any of these proteins have the capacity to undergo condensation on their own, similar to RNA-Z. Interestingly, we found these two proteins, HFQ, an RNA chaperone, and RHLE, a dead box RNA helicase involved in ribosomal RNA biogenesis, forming condensates in the presence of RNA at physiological concentrations and also in the cells. These are the program predictions for the liquid-liquid phase separation of proteins. RNA-Z is the positive control here. Intriguingly, no program predicted HFQ to be undergoing condensation. Since these two proteins are highly enriched in the BR bodies, based on both mass spec data and as well as the in vitro recruitment assay, we were curious to see if these condensates are part of BR bodies or can they also exist as independent condensates. For that, we depleted RNA-Z, which is under xylose-inducible promoter. Depletion of RNA-Z also depletes the BR bodies and thereby disturbs the association of BR body interacting proteins. Here I'm showing the mislocalization of aconitase upon BR body depletion. Similarly, HFQ condensates are also depleted when RNA-Z was depleted from the cell, suggesting that even though HFQ has the capacity to undergo condensation on its own, these condensates are mainly part of BR bodies. Interestingly, RHLE condensates still persisted even after RNA-Z was depleted from the cells, suggesting that RHLE condensates may exist as distinct condensates. When we looked at the colocalization of RHLE condensates and BR bodies by fluorescence correlation, there are homogeneous condensates where there is 100% mixing of both the proteins. And there are heterogeneous condensates, meaning that the concentration of RHLE is not the same in all the BR bodies. In vitro, when we mix the preformed RNA-Z condensates and the preformed RHLE condensates did not mix with RNA-Z condensates, suggesting that they can exist as independent condensates, and at the same time, they can also mix with BR bodies. Moving on to the final part. In my previous slides, I have mentioned that RNA-Z requires RNA for its condensation. Interestingly, few interacting proteins can also stimulate RNA-Z condensation. To understand which is the better stimulant of RNA-Z condensation, we perform time-lapse microscopy experiments and monitor the assembly of condensation. RNA-Z CTD doesn't form any visible condensates over this time. RNA readily induces the condensation of RNA-Z, and it is happening in minutes. PNPase induces RNA-Z condensation, but it's a very slow process. Addition of both RNA and PNPase has an additive effect. This combination increases the rate of the condensation reaction and also the number of condensates formed. The diffusion coefficient of RNA-Z in the only protein condensates is 3 times lower than the diffusion coefficient in the complex when RNA is present, suggesting that RNA may also increase dynamics in the condensates. And we know in the cells when transcription is inhibited by adding rifampicin, BR bodies are majorly dissolved, suggesting that RNA is required and it stimulates the kinetics of BR body assembly. To summarize the entire part, BR bodies contain diverse set of proteins, not only the mRNA decay factors. Though RNA is the central scaffolding protein, the proteins present in BR bodies can also condense in the presence of RNA. I have shown that HFQ condensates are mainly dependent on BR bodies, whereas RHLE condensates could be forming heterogeneous condensates. There are proteins which can dissolve these BR bodies. Here I have shown that S1 protein dissolves BR bodies by sequestering RNA from it. This could be an example of how strong RNA binding proteins can modulate the phase behavior of RNA-dependent condensates. The core degradosome proteins are localized to BR bodies. Few of these proteins can stimulate RNA-Z condensation in the absence of RNA, but RNA is an efficient stimulator of RNA-Z condensation, and it's also required for the assembly of BR body in the cells. I thank my supervisor, Jared Schrader, for the excellent project and guidance, and all the people who contributed to this project, especially Imolka, who performed in vivo experiments, all the collaborators, and NIH for the funding, and RNA Society for the research presentation fellowship, and Wayne State Graduate School for the travel fund. Thank you.